Let's provide a few examples of various shapes of nuclei that we can find in histological specimen. The shape of nucleus usually corresponds somehow with the shape of cell. So no wonder then that in flat cells we can have flat nucleus. Such as in squamous or flat cells. Spindle-shaped cells have usually spindle-shaped nuclei, such as in fibrocyte, where one of the edges would be usually sharp and the nucleus would be very dense. So it's called fusiform nucleus, such as in fibrocyte. That's a connective tissue cell. Similarly, in smooth muscle cells, for example, they are cigar-shaped or bacilliform nuclei with more oval or obtuse shape. Bacilliform or cigar-shaped nucleus in smooth muscle cells. Cuboidal cells have usually a spherical nucleus. Spherical nucleus in cuboidal cells, while columnar cells, those with one dimension exceeding the other dimensions, also the nucleus is usually elongated in the same direction. So you can call it ovoid nucleus in columnar cell. Other characteristic shapes of nucleus might be, for example, in blood monocytes, which are the largest cells of the peripheral blood, there is this incision making the nucleus look like a bean or a kidney. Kidney-shaped nucleus in a blood monocyte. While in other blood cells, the nucleus might be fragmented or divided into more lobes. So, for example, in neutrophilic granulocytes, the nuclear content is divided into more segments. Segmented nucleus. I will add the neutrophilic granules. Well, I'm ignoring the cell content in most other cells, but in this example I will add some, because this is a specific cell called neutrophilic granulocyte. Well, actually the number of the nuclear segments has some relevance to medicine, because young forms of the same cell have this nuclei that look like rod. So it's a rod. And during aging, the number of the nucleus gets more and more segmented. So there could be 1 up to 5 nuclear segments, and the number of the nuclear segments tells you how old the cell is. So both these are neutrophilic granulocyte. This one is a young one, this one is an older one. Some other cells, like eosinophilic granulocytes, have the typically bilobed nucleus. In this case, I will add the large reddish eosinophilic granules, so we will remember that the eosinophilic granulocyte has got these 2 nuclear segments. By the way, if I go back here, another term for this young neutrophil is stab cell, the one with the rod-shaped nucleus. The nucleus could have diverse irregular shapes, such as in macrophages. Macrophages are mobile cells that perform phagocytosis, and the nucleus might have variable shapes. So we have multilobed or irregular shape of nucleus, such as in this tissue macrophage. Or in megakaryocyte, which is a multinucleated giant or really large cell with many nuclei, but they frequently occur in chains, so it appears as more nuclear segments in a megakaryocyte, which is the mother cell of thrombocytes. But in this case, it's more nuclei, okay, in a chain. Let's consider various density of the nucleus and nuclear content. The nuclear content is also called chromatin. And the 2 extreme forms are the euchromatin it's a optically light form of chromatin. You don't see any larger structures there. The DNA is despiralized with a low condensation, is accessible to transcription enzymes. It prevails in interface cells, so mostly in the interface, and it appears as light regions of the nucleus, unlike the hetero chromatin, which is highly condensed nuclear content that provides some contrast in routine histological sections. It's basophilic, so it's dark, highly condensed, and it's mostly inactive in transcription. So starting with one of the extremes, the nucleus could look like optically almost empty. Perhaps you can see nucleus or nucleolus or more nucleoli. If it's empty, so it's mostly made of euchromatin, there's a good contrast showing the nucleolus or nucleoli. This can be found in some stem cells. Another example would be similar, still a nucleus made mostly of euchromatin, but in this case the chromatin is condensed below the nuclear membrane, and we call it vesicular nucleus. Vesicular nucleus, such as in some neurons or serous glands. Specialized for producing proteins. Most cells are somewhere in the middle, so you can see various patterns of a heterochromatin and the light euchromatin. So that would be nucleus of medium density in most cells during the interphase, while other cells have very characteristic chromatin pattern, such as this, where the position of nucleus is eccentric, and the heterochromatin, the dark component, forms like this clock face pattern or cartwheel pattern. This is in plasma cells, so you call it clock face or cartwheel pattern. And this occurs in plasma cells, which are cells of connective tissues that are producing antibodies, immunoglobulins. Other cells might have very dark chromatin. An example could be cardiac muscle cells. So high density of chromatin, in cardiac muscle cells, or in mucus cells that are producing mucus components. The whole nucleus is usually compressed in the basal compartment and mostly consists of heterochromatin. So this is another example of the mucus producing epithelial cell as an example. And the most extreme form of condensed chromatin could be seen in sperm cells, in a spermatozoa. So here the flagellum would continue, but I'm focusing on the head of the sperm, where the nuclear content is heavily condensed in the most compact, in the most possible compact way, and this is called pycnotic chromatin or pycnotic nucleus. That's the head of sperm cell. Also, during the cell death, the nucleus can have various forms. And if you can read this information from the slide, it could be useful. So the nuclear morphology during cell death. First, sometimes the nuclear content, the chromatin, starts to condense below the nuclear membrane. This is called marginalization. yeah, inside the cell. The highly compact form is called pycnosis, so the nucleus is very small and the nuclear content is extremely condensed, so this is called pycnosis. And it could precede the formation of apoptotic cell bodies. So during the programmed cell death, the apoptosis, we got more apoptotic bodies or fragments. With the fragments of the pycnotic nuclei, it splits actually. Or there could be destructive fragmentation of the nuclear content. So you can see some rough fragments and very fine dust from the chromatin. This is called karyopyresis, and you can see the destructive nuclear fragmentation, and the nuclear membrane ruptures. Another example would be karyolysis, where at first the nuclear membrane persists and the nucleus is decomposed via endonucleases. But the nuclear envelope persists. For some time. The reality in normal histological sections is much more complex. So this is like the first attempt to facilitate your contact with the various forms of nuclei, density of chromatin, and good luck with that.